Welcome, everyone. I'm excited to kick off today's presentation on tools of genetic engineering. This is the first slide, and it's our cover slide, so I want to briefly introduce what we'll be exploring. We'll look at the key tools used in genetic engineering and how they help us modify and understand DNA. We'll cover some fundamental techniques and their applications, all in a concise and engaging way. So, let's get started and dive into the fascinating world of genetic engineering tools. Let's take a closer look at the molecular tools of genetic engineering. Now, just like an engineer designs and constructs bridges, canals, and railways, a genetic engineer works with DNA to create and manipulate genetic material. The term genetic engineer is quite fitting because it describes someone involved in the precise manipulation of genes. Think of their toolkit as a set of powerful enzymes. These are the key players in recombinant DNA technology. These enzymes allow us to cut, join, and modify DNA molecules. They are the most commonly used tools because they give us the precision needed to work with genetic material effectively. So, understanding these molecular tools is essential because they form the foundation of all genetic engineering efforts. They enable us to unlock the potential of DNA, paving the way for advances in medicine, agriculture, and many other fields. This slide highlights the core components that make genetic engineering possible, connecting directly to the overall theme of how we can manipulate genetic material to benefit society. Restriction endonucleases, also known as DNA cutting enzymes, are some of the most important tools we have for manipulating DNA. These enzymes originate from bacteria, and their natural role is to cut or split DNA molecules at specific sites. Interestingly, they were first discovered in E. coli, where they serve to protect the bacteria from invading viruses called bacteriophages. The way they do this is by cutting up the viral DNA, preventing the virus from replicating inside the bacteria. It's important to note that the bacterial DNA itself is protected from being cut by these enzymes because it is methylated or modified at certain sites. This methylation essentially shields the bacterial DNA from cleavage. Because of their ability to cut DNA at precise locations, these enzymes are called restriction enzymes or restriction endonucleases. They are essential tools in genetic engineering and molecular biology research, allowing us to cut DNA molecules into manageable pieces for cloning, analysis, or further modification. So, in summary, restriction endonucleases are bacterial enzymes that recognize specific DNA sequences and cut at those sites, playing a crucial role both in bacterial defense mechanisms and in the techniques we use to study and manipulate DNA today. Let's take a closer look at the different types of endonucleases. These enzymes are crucial tools in molecular biology because they can cut DNA at specific sites. The main types we deal with are restriction enzymes, which recognize particular sequences in DNA and cleave at or near these sites. These are incredibly useful for genetic engineering and cloning. Then there are also site-specific endonucleases used in various DNA editing applications. Understanding these different types helps us appreciate how versatile and powerful enzymatic tools are in manipulating genetic material. This knowledge connects directly to the broader theme of how we can precisely modify DNA for research and medical purposes. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's talk about recognition sequences. These are the specific sites where DNA is cut by restriction endonucleases. These enzymes are quite selective. They recognize particular sequences of 4 to 8 nucleotides and then cleave the DNA precisely at those sites. What's really interesting is that each recognition sequence has what we call twofold rotational symmetry. This means that the same nucleotide sequence appears on both strands of the DNA, but in opposite directions. These symmetrical sequences are known as palindromes because they read the same forwards and backwards. Recognizing these palindromic sequences is crucial because it explains how restriction enzymes identify their target sites with high specificity. Connecting this to the overall theme, understanding these recognition sequences helps us grasp how enzymes can precisely cut DNA, which is fundamental in genetic engineering and molecular cloning. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's talk about cleavage patterns. The majority of restriction endonucleases, especially the well-known type II enzymes, cut DNA at specific, defined sites within their recognition sequences. This precise cutting is what makes them so valuable in genetic research. When these enzymes cut the DNA, they produce fragments that can have either sticky ends, also known as cohesive ends or blunt ends. Sticky ends are particularly useful for recombinant DNA experiments because they are single-stranded overhangs that can easily pair with other DNA fragments that have complementary sticky ends. This property allows us to join different DNA pieces together more efficiently. On the other hand, blunt ends are straight cuts across both DNA strands, which can be less specific but still useful in certain situations. The key takeaway here is that the ability of restriction enzymes to generate sticky ends enables us to manipulate DNA more effectively, especially when constructing recombinant molecules. This concept ties back to our overall theme of understanding how enzymes help us work with DNA in precise and predictable ways.